Hello and welcome to the SCI Guys, the show where we talk about the crazy, weird, and wonderful stories from the science world. I'm Corey, and as always, I'm joined by my co-host, Jeb and Luke Gutforth.
Hello!
Hi, howdy.
This week we're talking about stolen science.
Ooh.
But first, we've got to do a little YouTube comment. So this one is actually from the man who drank radioactive water until his jaw fell off.
Oh, yeah.
The comment says, "Hello, from the Spotify podcast. I've had too many x-rays for my 2 broken legs, 1 broken spine and thumb. Not to mention 4 MRIs and 1 CT scan. And I don't know how much dental x-rays. Love your episodes. See ya."
Goodness me, I hope you're okay.
Yeah, I know.
Are you okay?
So many broken bones. Oh my lord. Wow.
I was thinking so many x-rays. Like x-ray exposure.
Yeah, but 2 broken legs and a broken spine. I feel like...
They're not been able to go anywhere. They're just listening to us all the time.
Exactly, right? Yeah. That's what we should do. We should break people's legs. They have no option but to listen to us. Comment on the YouTube from Spotify to answer this question. Would you have a company like 23andMe, for example, sequence your DNA? Would you do that? I don't know. Would you do that?
Well, you've told me multiple times not to do it, so apparently not.
Why don't we start the episode?
Spoilers!
Actually, no spoilers at all. So I want to talk today about DNA, the discovery of DNA, because I feel like people don't know very much about the discovery of DNA. They know broadly some parts of it, but not really the ins and outs, if that makes sense. Mm-hmm. So first off, before we get into talking about the discovery of DNA, we need to know what DNA is. So what's DNA?
Deoxyribonucleic acid.
That's
it.
Close, yes. Deoxyribo.
It's a little spinny thing.
It's the ladder that makes you who you are.
Exactly, yeah. So deoxyribonucleic acid, yeah, absolutely. So from Britannica, it says DNA, abbreviation of deoxyribonucleic acid, organic chemical of complex molecular structure that is found in all prokaryotic and eukaryotic cells and in many viruses. DNA codes genetic information for the transmission of inherited traits.
Yeah, the ladder that makes you who you are, just like I said.
Yeah. I mean, the one thing that you didn't mention is that we find it pretty much across almost all life that we're aware of, which is pretty cool.
Almost all.
But yeah, not all viruses have DNA. They can have RNA.
Oh, okay,
yeah, exactly.
Okay.
You got it?
Yeah,
you got it?
Distinction.
Yeah, exactly. So, DNA. What is the structure of DNA? What do you know about DNA? Give me some more facts about DNA.
Double helix. Spiral staircase, isn't it?
Yeah.
Yes.
So it's not a staircase.
It's the spiral
ladder.
staircase that makes you who you are.
It's not a staircase.
What is a ladder if not a staircase? It's just a stripped back staircase.
Well, I'm not going to get into
staircase.
That. It's made up of 4... Oh, what are they called? Flipping heck. Oh, what are they called? Base pairs.
Yes.
No, yes.
Well, 2 base pairs.
2 base pairs.
Yeah.
4 bases. Okay,
yeah.
Thank you. And those are written out by us as A, G, T, and C.
Exactly.
They code for proteins. Proteins make up cells. Cells make up you.
Yeah. Pretty spot on.
Good summary.
So DNA, as I've said, it's got a double helix. Well, as Jamp has said, it's got a double helix structure. It's a polymer. It's a spiral. It's got 2 DNA strands in sort of each double helix. Yeah?
Yeah.
You got 2 strands of DNA in your DNA. That makes sense.
Yes.
Your DNA. So when we talk about the double helix, that's 2 strands of DNA, right?
A
Twirling
core
around
DNA
each
is
other.
actually 2 DNAs.
Yeah. Exactly. Yeah. Cool. Sure.
Yeah.
So the nucleotides of DNA. So we'll talk about what that is in a sec. So that is deoxyribose sugar. That's the deoxyribo from the deoxyribonucleic acid. And that is joined to a phosphate group and then one of the 4 bases that you mentioned there, Luke. And do you know the names of those bases?
No.
I was going to ask you to name them when you said, we write them out as...
I remember them trying to get us to remember them in biology A level, but no, I cannot remember them. I'm sorry.
So they are... Well, they're split up into 2 different categories. They are the purines and the pyrimidines. The purines are adenine and guanine, and the pyrimidines are cytosine and thymine.
Yeah.
And so A joins to T, so adenine joins to thymine, and guanine joins to cytosine. Yeah? Yeah. And with that, you code basically everything. Every single protein you've got can be coded by those 4 bases. Pretty cool, I think. It's pretty insane.
They use lots of them.
Yeah, exactly. Yeah. And DNA is pretty consistent. We were speaking about this a few weeks ago. DNA is pretty consistent across pretty much all, like to the point where, we could take a bacteria. Yeah, we could take some bacteria and we could take some genes from our cells and be like, okay, here you go, bacteria. Please make these proteins. So the bacteria will just make the proteins.
Could you expand for a moment on the idea of what you said about adenine binds to guanine? Is it?
Thymine.
Thymine.
Yeah.
And then the other one binds to the other one.
So-
What do you mean by that?
Okay, so you've got the way that it goes. So we've spoken about the double strand.
Yeah.
Right. So the way that it works is that there's nucleotides, and those are the different sort of segments of the ladder, right? If we think about the steps of the ladder, those would be your base pairs. So your adenines and your thymines and your guanines and your cytosines. Yeah?
Yeah.
Yeah.
So those would be the steps of the ladder because they join together. So adenine bonds to thymine to make the step of the ladder.
Right.
So, one strand of the DNA will have an adenine and the other strand of the DNA will have a guanine. They will bond
see.
together.
Okay.
And they'll make the sort of steps of the ladder.
So it's the binding is how they join the two separate DNAs together.
Yeah, the two strands of DNA are joined by... So they're kind of... The two strands of DNA are... They're kind of complementary to each other, right?
Yeah.
So if you've got... Gosh, this is kind of difficult to talk about without visual.
Yeah.
So if you've got the sort of there's two different ends of DNA. You've got the 5' end and the 3' end. You don't need to worry about what that means. Just know that one end of DNA is 5', the other end of the DNA strand is 3'. Now the way that it goes is each strand goes. So the strands that join together, one will go 5' to 3', and the other will go 3' to 5'. And they're complementary. So the sort of, I guess, the code of the DNA, right? The code of DNA, when we talk about the code of DNA, that is literally just the order of A's, G's, C's, and T's.
Yeah.
Right? So your DNA code is just whatever order those are in. So one strand from the 5' to the 3' end might have A, C, G, T.
Yeah.
Right? And that means that the other strand of DNA from the 3' end to the 5' end will have T, C, G, A.
So if you were to start at the top of DNA, strand 1, and you were to go all the way down, and then you start at the bottom of strand 2, and then go all the way up, that would be a palindrome.
No, because the bases are complementary, right?
Okay.
So the way that it works is essentially you've got two strands of DNA, right? And it doesn't really matter for what we're talking about today what 5' or 3' means or what ends those are. All you need to understand about DNA is that one strand is complementary to the other strand, by which I mean the bases on one strand match up to the bases on the other strand, right? Because they need to bond together and they can only bond to one base can only bond to a specific other base.
Yeah.
So you can always tell what the code of the other strand is based on the code of your first strand because they have to be complementary to join together.
Yeah.
And an additional thing, I mentioned the 5' and 3' ends, that's not super important for what we're talking about today at all. It's not even really super important for your understanding of DNA. But the reason I mention that is because it's important when we're talking about DNA. So when you're working with DNA, it's important which sort of way the strand goes because they're not sort of symmetrical, right? They run from sort of 5' to 3', let's say. And that's just an important distinction. You don't need to understand really what that means. Think of it kind of like an arrow, right? It's important to know which end of the arrow has the arrowhead.
Yes.
Right? And with the two strands of DNA, one of them has the arrowhead pointing, say, to the left, and the other has the arrowhead pointing, say, to the right. Right?
Right.
Does that make sense? And that's all you need. That's all you need to know when you're thinking about it, right? So if we were to think about it, actually, let's use the arrow thing, right? So you've got one arrow pointing to the left and one arrow pointing to the right, and you're say writing letters on the two arrows, right? If you write A on the top arrow, you need to write T on the bottom arrow.
Yeah.
Okay. So the top arrow is pointing to the left, the bottom arrow is pointing to the right. Now, if you were writing sort of A and then writing a T on the bottom arrow and then writing, say, a G and then writing a C on the bottom arrow, you're not going to have the same, like, if you lie the arrows next to each other, both with the arrowheads pointing in the same direction, you're not going to have a code that matches.
Yeah.
It only matches when the arrowheads are pointing in the opposite direction. Mm-hmm. That's the same with DNA, essentially, right?
Yeah.
Like that's all you need to know. You don't understand why that's important or whatever. That just explains the 5' and 3'. There you go. You understand DNA a little bit more now.
Perfect. I'm a geneticist.
Close enough.
To a degree.
What do geneticists do? No, I'm joking. They do a lot. So, DNA, when I say it's a polymer, do you know what I mean by a polymer?
No.
So, well, you kind of understood the concept already. It means that it's a long chain made up of individual units.
Yeah.
And each unit is that sort of sugar connected to the base pair. So you've got your sugar with your phosphate, which is just like, sugar is a kind of, you know what sugar is. Mm-hmm. It's a kind of chemical. A phosphate is another kind of chemical with phosphorus. and your base pairs are your A's, C's, G's, and T's. So if you take a phosphate, the sugar, and the base pair, which is an A, C, G, or T, that is your individual unit. DNA is just made up of a long strand of those individual units. Yeah? And as I've said, they're complementary to the other strand. They match up, they link together. That's how we get our code, right?
So insane.
It's so insane. It's like once, this is the thing. I've obviously studied this from when I was, they start you when you're in primary school, right?
Yeah.
And I just continued that throughout high school, then I did it into university. So it just makes sense. It's very cool.
Yeah.
It just makes sense. As soon as you like sit down and like, if you sit down and try to learn it, like if you were to look at the sources that we've got and sort of connected to this episode, you could very easily understand the general concept of DNA. Like the concepts surrounding it are not terribly difficult. It's all pretty straightforward and really cool. When you start to get into the nitty gritty, it's very complex. Like it's incredibly complex, but you can simplify it down to a way where it's really easily understandable, because at its core, it is just some letters that make a code. And then you translate that code into proteins. Like it doesn't matter how you translate that code, but you just translate the code into proteins and then that's it. Like it's very straightforward. It's so simple in concept. It's amazing to me. So moving on, that is DNA. That's basically what DNA is, right? So it's you understand that it's sort of individual units making up a long chain, and certain bases can only bond to certain other bases. A to T, G to C. Yeah?
Yep.
Awesome. So now, how does DNA work? Like, what's its job? What does it do? Why is it good at its job? Why do we need it to do what it does?
That's a question for us.
Sure, you can answer any one of those you want.
Isn't it?
Just generally.
We need it to make our bodies.
Yeah, exactly. Yeah. We need to make our bodies. Like, why? What is it? What does DNA do? Just sort of generally, like, what is its general function? You don't need to know the answer to this.
Sustain all of life.
So, kind of, its general function is to store data.
Yeah,
it's a data store,
okay.
right? Because your DNA isn't. If we're being sort of really specific, your DNA isn't actually what makes the proteins, right? Sort of directly. Right, it's what codes for the proteins. It's your data store. It's like your sort of it's your hard drive, right? It's where you store all of that information, but you don't make it. You don't sort of make your proteins directly from your DNA. There's some intervening stuff there. Yeah. So the reason that DNA is really good at its job is because it's a really stable molecule.
Yeah, I was going to say it's very resilient.
Exactly.
Because there's people looking into using DNA to store data, like encoding our data from like a computer or whatever that you want to have last for a very long time, because I don't think we really think about the fact that, like, you know, when you have like the burning of the Library of Alexandria, like you have information that, like, we think because we have the internet and because we're not responsible for looking after our data a lot of the time because we buy a phone that does it, or we put it in the cloud and somebody else does it. If you lose data, that's it. It's gone. No one will ever know it ever again. And if it's not in a person or it's not written down, they have to rediscover it, which is like ridiculous. I don't think we as normal people really conceptualize of the world in that way, because it's so obvious that, well, someone knows the answer to that, but only if they exist and only if it's written down. And all the ways that we have of storing information, like hard drives or floppy drives or like USBs, they all break down eventually. So we're trying to find ways of storing information that will last thousands of years, and we don't currently really have that.
Well, it's hard. It's hard to make anything last that length of time. And the thing with the way that we store data and the way that we store that, it's all very small. Do you know what I mean? In that it's all very, very precise.
Yeah.
And being very small and very precise kind of means that over a longer period of time, any small change or any small problem or issue is going to affect the data, right?
Yeah.
Because, you know, if you're very, very precise and you just change it just a little bit, boom, data's
especially if you've encrypted it.
Yeah,
if
exactly.
You've encrypted it and you lose one little bit. I don't know quite what the fault tolerance of encryption is, but there'll be some forms of encryption where you lose one bit, 1 1 or 1 0, and without guessing what those bits are, it's gone.
Yeah, it's insane. And I mean, when you think about DNA is pretty good. I mean, obviously there are mutations. It's not perfect in terms of it's not a perfect store of, I guess, information or data or whatever, right? It doesn't store it perfectly, but there are sort of things in your body and things in other creatures' bodies, other living things' bodies, that essentially kind of fix DNA. So you've got you replicate DNA, and that could there are mistakes when there could be mistakes when you replicate DNA, which causes mutations, which is the reason for ginger people existing, for example, or for paler skin existing,
or
right?
for anybody existing, really.
Well, yeah. I mean, and this.
But also ginger.
It's one of those things where it's not a bug; it's a kind of a feature, right? DNA replication isn't 100% perfect. You don't store the information absolutely perfectly, but that's beneficial, or it can sometimes be beneficial.
I'll go back in time to high school and tell all my bullies that being ginger is not a bug, it's a feature.
Ooh, buddy.
Excuse me. Being ginger is not a bug, it's a feature. Ah! Ow!
If you're a ginger kid, I promise you, do not say that to your bullies. It will not help. You will get bullied. And put it this way, don't take advice from any of us on how to not get bullied. Because we don't know.
We'll have to bring in an expert for that.
My advice is become an adult. That's it.
Yeah.
That's it. Become an adult.
And it kind of flips when you become an adult.
Yeah, that you start bullying kids.
No.
Yeah.
Well,
no,
you
no,
can.
That's really
what are they going to do?
That's not what I meant, though.
The science of not being bullied coming soon.
So yeah, DNA is essentially the store of your data, the data that makes you you. It stores it. Now, when you want to make new DNA molecules, obviously you replicate the DNA. There could be mistakes there. We've mentioned that. Those cause mutations. Mutations can be good or bad. Realistically, a mutation is neutral, but it can confer a benefit to survival, or it can be a hindrance to survival. And that's kind of the basis for natural selection, evolution, all of that sort of stuff, right? And when it comes to turning DNA into proteins, that's where RNA comes in. We don't need to worry about what RNA is, but to turn DNA into proteins, you use RNA kind of as in the middle, right?
Yeah.
Does that make sense?
Yeah.
Yeah?
Yeah.
Yeah?
Yeah.
Yeah.
Sure.
Do you want to know how it works generally?
Yeah, go on.
Yeah.
Okay, so basically what happens is you kind of split apart the DNA strands, right?
Unzip.
Yeah, unzip them, right?
Oh.
And then you got the RNA comes on in, the messenger RNA comes on in, and it connects up to the DNA strands. Because obviously, think about it this way, right? These are individual units. So all you need to do is have an RNA with, your G. You've got a G there sitting on your DNA strand. An RNA with a C comes on in.
Yeah.
Boom.
Great.
Connected to you.
Fantastic.
And then, it just goes down the line. And then all those RNA strands just get connected. All those little individual RNA bits get connected into a whole strand once they've sort of bonded with the DNA.
Yeah.
Right.
Oh.
So now now that's the same as the DNA code.
Yeah, but
And
it's
then-
made a different thing. Yeah.
It's a slightly different thing. It's RNA, which
So that's
isn't
a copying process.
yeah, that's the that is a kind of a copying process. The RNA isn't as stable as the DNA. Then the RNA goes somewhere else, and that's used to code directly for proteins.
Right. So that's sort of in sort of a more accessible idea. That's sort of like copying. You got your like deep data storage, like all the deep data storage we use is like on tape, for example, and it's really slow. And you might copy that onto a flash drive.
Yeah.
And in order to plug it into your computer.
Yeah.
Because your computer can read a flash drive, but it can't read off of tapes. But the flash drive is dodgy, and it might break, and you might lose it, and but it's fast, but it's breakable. So then you can copy it onto your computer to then open up your picture or open up your video or whatever.
Yeah.
So it's that copying device.
Absolutely, yeah, spot on. It's essentially, right? That is not the full picture. It's more complex than that. It's biology. I'm not going to be able to describe it quickly within the space of a couple minutes.
Can I just check? So that RNA then goes off to the sort of protein building apparatus, and is read and then builds proteins. No. Okay.
Yes.
I took it too far.
No. Yes. The RNA goes to the protein building apparatus, your ribosomes, which is just it's a thing that's inside of
Turing
your cell.
machine, isn't it?
How do you mean?
There's a little inside of the cells. There's a little Turing machine that sort of reads it and then makes a thing.
Yeah. So there is another sort of intermediary as well.
Sorry, I'm
kind
making
of
this more complicated.
No. We've spoken about codons before. So
Yeah.
codons basically what they are is the sort of you've got three base pairs. So you've got, let's say, an A, a C, and a G all in a row, right? That is one codon, and one codon will then sort of that's matched up to one of the amino acids, which is a building block of protein.
Yeah.
Or a codon could mean stop here.
Yeah.
Protein
This is-
finished.
And this is exactly how a Turing machine works as well, which is so cool.
Yeah.
It's
right,
just
well.
like a little tape head that's reading, like, rather than read it, it's either reading like punch holes in a card, or it's reading code off of a piece of tape, and it's just going along and it's just reading, and then that whatever it reads corresponds to an action, exactly like
Yeah.
pulling out an amino acid.
Yeah, exactly.
Oh, it's so freaky.
Exactly.
We
It's
invented
crazy.
we invented a Turing machine, and then we found out that that's what our cells are doing.
We just copied ourselves by accident. Yeah.
Oh,
right?
that's so cool.
So, what this is, there's not a huge reason to go into this. I just find it kind of interesting. But yeah, so that's kind of how DNA becomes a protein.
Sorry, imagine if we like invented the iPhone, right? And then when we invent a microscope and we look inside of like say a quark, and we're like, well, the quark's the smallest thing, and then we look inside the quark, and inside the quark is loads of iPhones. That'd be so weird.
That's crazy. But we spoke about Velcro a few weeks ago. Like it's called biomimicry. We take often it happens intentionally, but we just take things from nature.
Yeah.
And a lot of the times nature will find the simplest way to do something. Now, don't think that the simplest way to do something necessarily means either the best way or the absolute simplest way you can think of. What I mean by that is that it will use the resources that it's got to, you know what I mean? Like it's not going to like when it comes to evolution and all of that sort of stuff, you're not going to have necessarily an overly complex way of doing something. If there is an easier way of doing it that requires less energy, and you already have all the sort of stuff to, you know, all the apparatus to make it.
And also I think what you're getting at there is that the simplest way of doing something also depends on what nature's already done. So nature is not going to undo a bunch of stuff and then go back to first principles and redo something. It's going to do it the simplest way, or one of the simplest ways, based on where it's already got to.
Yeah.
Which means that you can get these sort of weird solutions that there would actually be a much better solution, but because it's working with what it's already built, it's the best solution with the apparatus it has.
For example, let's think about bats, right? Now, is a bat's wing necessarily the simplest way to make a wing?
Perfect. No.
Make all your fingers really long and your skin really thin and webbed between them?
Maybe.
Is that the simplest way of making a wing?
No.
Or are birds'
Could
wings
be.
the simplest way of making a wing, where your arm gets all messed up and jacked up and your fingers become long and they go like this and bones fuse together and you got this?
Or an airplane wing.
Or an
You just
airplane
have a
wing.
shape that
Exactly.
manipulates air as it
Or a
passes
paper
over.
aeroplane wing.
Exactly, right? Now, these
That's
are all varying degrees of simplicity. Genuinely, these are all varying degrees of simplicity with wings. Now, we could point to one of them and say that's the simplest way. But it's not the simplest way necessarily, because to make a paper airplane wing from a bat wing, from like what a proto bat would have had, you would have had to have made a lot of changes. But to make a bat wing from a proto bat, just make its fingers longer.
Also, very interestingly, it's interesting that we call both those things wings, because they don't, like an airplane wing and a bat wing are totally different things. One is forcing air down and then creating a vacuum above it, which sucks up. So one is forcing air down by pushing it down and then creating a vacuum which pulls up. And the other one is manipulating airflow with a forward direction that causes a vacuum above that sucks up.
Yeah, and also, we don't have, helicopter rotors are not called wings. And so wing doesn't necessarily apply to something that aids in flight, a long, relatively thin thing that aids in flight.
Yeah.
It's just, I don't know, what is a wing then?
What is a wing?
What is a wing?
Let us know down below.
Yeah.
I think wings generally would have to be on either side of like a thin object. So like a bird has a wing because it's on both sides. And then a airplane has wings because they're on both sides. But a rotor rotates, and it's not. It's so weird. Sorry, we're massive tangent.
Well, there's fixed. I guess it's the distinction
wing,
between fixed wing
and flapping wing.
Yeah. So wings, if we're going to Google it, in a bird, a modified forelimb that bears large feathers and is used for flying, and a rigid horizontal structure that projects from both sides of an aircraft and supports it in the air.
Both sides.
Bats don't have wings. Based on these two definitions.
Oh, okay.
Yeah.
Well, it's semi-rigid. They're semi-rigid.
Right. But bats' wings don't have large feathers, but they are used for flying.
Yeah.
So they need to have large feathers.
So I suppose a wing could be a modified forelimb used for flight.
Yeah.
Yeah.
All right. Or, in regards to man-made stuff, it's rigid structures on either side that are horizontal, used for flight.
Yeah. Interestingly, on your point when you were saying about evolution won't necessarily find the simplest way of doing something, a really great example of that is that evolution hasn't got a lot of examples of, for example, like you say, like motors, like which happens with a helicopter, but it also doesn't have a lot of examples of wheels. And that's really interesting. And there's a load of studies on like whether a wheel could have naturally invented, and it's, I can't remember exactly why, but there aren't many. There are examples of motors like on the tiny scale. We have little biomotors that happen very small scale. Yeah. But on a large scale, there aren't really natural motors. That's something we genuinely seem to have invented.
Well, there's a reason for that. In that wheels would require for something to be fully, like a full rotation, and for something to be kind of detached, right?
Yeah.
That is kind of difficult to do in nature. Now, I think we've spoken about this before, but in his Dark Materials, Philip Pullman's novel series, The Golden Compass, if you've seen the film, the books that's based on. Now, there are these sort of, I guess, not interdimensional, but extra-dimensional beings from another universe, let's say, that are kind of like elephants, but they've, I guess, evolved in a completely different world, a completely different planet, completely different universe. And what they do is they hook their feet through these big seed pods that have holes in the middle, and they essentially ride around on wheels, right? There's these like super intelligent elephants super intelligent for elephants. They're slightly less intelligent than humans. And they sort of hook their feet through these like little seed pods and use them as wheels. That's like a biological wheel. Yeah. But one of the reasons that a wheel is really difficult to evolve is that a wheel would kind of need to be separate from the rest of the body, and then that's difficult to maintain.
But could it not be like an owl's head that can rotate 360 degrees? Could have owls' heads on both sides of your body. I think as well.
They can't like continually.
Yeah, I don't think they can continually rotate. Those can just. What it means is that an owl can just like.
It can go all the way.
It can turn its head entirely.
It can do
around,
one rotation, but you need to do multiple for it to be a useful wheel.
Yeah. So, and owls can rotate their necks a maximum of 270 degrees without breaking blood vessels or tearing tendons. That doesn't mean that they can spin freely.
Yeah.
And that's the thing. You don't really see free spinning things in nature because if you think about it, how the hell are you going to get energy to it? So you could potentially do it if it's, let's say, something like keratin, like hair or nails, and you grow it out and then it detaches and then it's sort of dead. That's fine. But then it would wear down. How do you sort of grow that?
Oh, yeah.
You need a blood supply to something to give it energy and, and oxygen.
Wow.
To replenish
it.
Yeah.
And if something's going to have a full rotation, it can't have a blood supply. So that would be why you can't sort of do that in nature.
That's so interesting. Another theory I read as well, which is probably supplementary to what you've said, is that roads are required for wheels. You can't have wheels on grassland, really. They're not as effective as legs. Or flying.
Yeah.
And so, you need to like sort of manipulate the surface in order to have wheels, really.
Yeah. Well, there you go. So there's like, obviously, this is getting more into evolution, but I find this all I find this all really interesting. I find this all really interesting. I think that you've kind of hit on it there. You don't have the selection pressure for wheels, by which I mean there's
not an opportunity.
Yeah, so wheels don't confer an advantage. So there's no selection pressure for you to have wheels instead of legs, especially considering to get to wheels, you can't just wake up one day and there's a chicken with wheels.
Yeah.
You need to have like sort of individual chickens that have closer and closer to having wheels. And sort of at no point before having a wheel is there going to be a benefit?
A helpful thing.
Yeah, exactly. Yeah. So there's no selection pressure there. But also, just physically speaking, it's biologically very, very difficult to sort of develop, right?
So interesting.
Yeah.
There we go.
Oh, it's great. It's great.
Anyway, that's that tangent over, audience.
Ding-a-ling-ling, is that the ad bell?
It sure is.
It's the ad bell.
Well, the ad gods have asked us to promote our Patreon on this episode.
Hell yes.
Thanks to our patrons over at patreon.com/Sci Guys. You get Sci Guys every single week, but if you become a patron, you can get a whole host of other things. What can they get if they join our Patreon?
An extra episode a month.
You can get access to our super secret Patreon podcast, After Dark. Just because Sci Guys has ended doesn't mean the conversation has.
That's right, and you can also vote on episode topics, submit episode topics for the vote, and some patrons can just get rid of democracy altogether and tell us exactly what episode they want us to make. You get bonus clips, behind the scenes stuff, lots of cool stuff over on our Patreon. You should definitely go and check it out right now at patreon.com/Sci Guys.
Don't miss it.
I agree.
Let's start the show again. So we've kind of gone over what DNA is, the structure of it, kind of the function broadly, a little bit about evolution. Another thing to mention is that DNA is organized into chromosomes in your cells, which you probably know what a chromosome looks like. It's kind of like a funky little X.
Little X,
Yeah, funky little X. Do you know why they're called chromosomes? Just a fun little fact. Do you know why they're called chromosomes?
No.
Right. So chroma. What would the word chroma
Colour?
mean? Color. Yeah. Right. Chromosomes. Now the reason they're called that is because when we discovered them, it was by staining them. So they're colored.
Oh.
What's the zomes mean? Do you know?
There's just some of them about.
Yeah. So
Some
is
colours?
We found some.
Chromosomes?
We found some.
inside your cells. There are a lot of different things that have a sort of zome suffix. I don't know what it means. I don't know where it comes from. Someone will probably tell you in the comments.
Great, please.
There you go. So we've spoken about all of that. Chromosomes, your funky little X's, that's how your DNA is sort of organized. And yeah, I mean, that is sort of an overview on what DNA is generally, right? We could probably do another episode entirely on DNA, replication, all of that sort of stuff, because there is so much content there. But I want to get into the history of DNA, the sort of discovery of DNA. Who discovered DNA? Do you know?
Crick Ann Watson.
Wrong!
No!
Absolutely wrong.
Was it a woman that was promptly shamed and kicked out of science?
Are you talking about Rosalind Franklin?
Yeah, that's what I mean.
Damn it.
Wrong!
Oh.
No! None of these people discovered DNA. Well, when do you think DNA was discovered? How about this? What century?
19th century.
18th century.
Yes, no.
19th century.
19th century. Absolutely. So it was 1869, but it took quite a while for us to understand why it was important, all of that sort of stuff. So in 1869, DNA was discovered by a man named Friedrich Miescher. He was a Swiss chemist. Now, this discovery wasn't seen as very important for quite a while, but I think it's really important to mention because we often think of science as being sort of one or two people doing great, massive
Eureka!
things. Exactly. People talk about a eureka moment and, one person just has a groundbreaking discovery because they are smarter and better than the rest. That's not how it works, really.
Also a little bit lucky.
Well, yeah, there's a luck element there, but also it's all built on the sort of the groundwork of so many other people. Like so many other scientists build the foundations for great discoveries.
Yeah.
You wouldn't have Watson and Crick discovering creating a model of the DNA of the structure
Helix,
of DNA.
Yeah.
Without the work of every single other person before that.
The inventor of ladders.
Exactly, the inventor of ladders.
Very true.
Exactly. Well done, Luke.
I hope they got some of that Nobel Prize money, Crick and Watson.
So the important thing to understand is that it's not a case of just one singular person making a massive discovery. I think it's really important to think about the fact that there are lots of people along the way that add to the sort of canon, add to the knowledge, and that is what makes it possible for others, to make these groundbreaking discoveries. So we're not going to talk about every single person that has worked on DNA, today. We cannot do that. But I think it's important to bring up the person that discovered it and that it wasn't Watson and Crick because people think that it was those two that discovered it, but it was actually discovered a lot earlier. So, as I said, Friedrich Miescher was the one that first identified it. He called DNA nuclein. He was looking inside the nucleus of human white blood cells. Essentially, what he did, he actually got those white blood cells from, I think, sick people from a hospital. What was it? He got them from, I think, bandages. Used bandages. Yeah.
Lucky.
So he was looking for something else pretty much entirely. Then he realized he discovered something new. And he said that, this was a phosphorus-containing substance. He said, an actual quote, "It seems probable to me that a whole family of such slightly varying phosphorus-containing substances will appear as a group of nucleons, equivalent to proteins." So that's him there laying the groundwork saying, yeah, these are going to develop and they're going to be the equivalent to sort of proteins, which was pretty much spot on, right? That is what DNA is. There are some different, there's different sort of variations of this, like A, C, G, and T. Different variations of the base pairs, and they do have a relationship with proteins. They are sort of equivalent to proteins. So he discovered the DNA molecule, right? Now we didn't know the structure of the DNA molecule. This is something that is sort of important to understand. We knew that DNA existed. We knew what it was kind of composed of. We did not know what the structure was. We didn't have the technology at the time to figure that out. So that was Miescher's discovery, right? And the issue was that for a long, long time, that just went unnoticed. No one really cared about it. So here is a quote, right? In a 1971 essay on the history of nucleic acid research, Erwin Chargaff noted that in a 1961 historical account of 19th century science, Charles Darwin was mentioned 31 times, Thomas Huxley 14 times, but Miescher not even once. which is insane because he discovered DNA, basically. Was not mentioned once in this sort of summary of discoveries from that time.
We're mentioning you, baby.
Well, that's the thing. We didn't know. It wasn't because they thought, oh, yeah. They just didn't know how important DNA was as
Didn't
a
they,
molecule.
like, at some point, like, sort of disregard it? They were just like, oh, I don't know what that is. It probably doesn't do much.
Yeah, we disregarded it. Exactly. And so then a lot of people did work over the years. And in walks, let's say, Rosalind Franklin and also Maurice Wilkins. So they were working at King's College London and they were using X-ray diffraction to study DNA. And this is where it gets to be a little bit of an issue. So these two were, they didn't publish their research, right? They hadn't published it. No, they hadn't published it yet. But Watson and Crick came across it. They were shown it by, I think it might have been Maurice Wilkins, but they were shown it by someone else, not Rosalind Franklin. And they then used that to sort of build their work on. Now, you could actually see, I'm pretty sure that in the X-ray crystallography from Rosalind Franklin and Maurice Wilkins, that you could kind of see the structure, essentially, right? As in, they didn't necessarily steal. They did add something to it, but their work was very much built on the work of these other two scientists.
Were these other two scientists still alive when Crick and Watson got their Nobel Prize?
Oh, so we'll get into that. They were alive when the paper was published, right? I'm pretty sure. And at the time, actually, Watson and Crick, there was someone else that was working on. There was someone else that was working on, looking for the structure of DNA at the same time. That was on a sort of similar path, and they were really worried that person was to scoop it out from under them,
like Darwin.
Yeah, exactly, right? So let's finish with Meischer for a sec and then skip forwards. We're going to move past a lot of other people that had worked on DNA over the years and move to Rosalind Franklin. So she was a great scientist. She did an incredible amount of work for finding the structure of DNA. She did a bunch of X-ray crystallography. She basically laid the foundations for Watson and Crick making a model of the DNA structure, essentially, right? So let's just go into a little bit of her life. She was born 25th July 1920 in London, and she essentially then studied at the Women's College at Cambridge University. She studied physics and chemistry, and then she went to work for the British Coal Utilisation Research Association. And then she moved to Paris in 1946, where she learned and sort of, she got those skills in X-ray crystallography. And now, she died quite young, right? She was apparently known to be passionate and kind of difficult to work with sometimes. She was very much someone that sort of spoke her mind. She, literally a quote right here, she had a great love of the outdoors and enjoyed spirited discussions of science and politics. So she she could be sort of short-tempered and, that kind of person. So now she was working on sort of looking for the structure of DNA. She was doing X-ray crystallography on the structure of DNA, which you don't need to worry about what X-ray crystallography is. Okay. Doesn't matter at all. It's just something that we use to figure out the structure of things. That's all you need to know.
In my head, it's like it's almost like you got some DNA, you just like shine a little torch on it, and the DNA is really small, but the wall you're shining it on is really far away, so you just see the little double helix as a shadow on the wall. You're like, wow.
You do a little puppet show with it.
Yeah,
Not
exactly.
quite.
Look at that.
Not quite.
There, that's the structure. There it is.
We'll talk about X-ray crystallography another time. But what to take from this is that she did this work. She had this unpublished data, and Franklin had no idea that Watson and Crick had seen her unpublished data.
Drama.
So now there was a photo 51 which was shown to Watson by Maurice Wilkins, who was someone that was working with Rosalind Franklin on the sort of looking for the structure of DNA. Now, this photo 51 was basically an image of a DNA molecule, and that was apparently the inspiration for Watson to sort of build the model of DNA as being the double helix, because apparently in photo 51, this photo of the structure of this photo of a DNA molecule, the helix was present. The double helix was visible, essentially.
Blimey.
Yeah, the pattern, it says here the pattern was clearly a helix. So
someone called Drama Alert.
Yeah. So they used
Welcome, Drama Alert Nation.
They used Franklin's data and then also her photo and used that to build their model and then did not credit her.
That is some beef.
Exactly. So Rosalind Franklin then moved to Birkbeck College. She was working on, other things. And then she died of, I think it was ovarian cancer, when she was 37 years old. So I think that was 1958. She died.
Goodness.
And then Watson and Crick, they went on to build that model of DNA, win the Nobel Prize, and took a little bit to actually acknowledge like the contributions of the woman that they just fully stole from.
Didn't Watson and Crick claim that they came up with the structure of DNA whilst tripping on LSD? I'm pretty certain I read that.
I feel like you've talked about this recently as well.
I'm pretty certain that's what they said. They said like, oh, the idea of it came to me whilst I was tripping on LSD.
It says here, soon after Crick's death, there have been allegations about him having used LSD when he came to the idea of the helix structure of DNA. While he almost certainly did use LSD, it's unlikely that he did so as early as 1953.
All right, I'm a source of fake news. My apologies. I retract my statement.
1953 was when they had they published sort of model, and that obviously led to them getting the Nobel Prize. Now, the model has kind of changed over the years. We've kind of made minor sort of adjustments and whatnot, but there are four main features that you sort of make up the model, and those are still consistent today. So DNA being a double-stranded helix, which that's the basic thing, right? Where you've got the A's and the T's joined together, and the A's and the A's and the T's joined together, and the C's and the G's joined together.
Yeah.
Right. That's part one. So it's a double helix joined by those base pairs.
Yeah.
Yeah.
Part two is most DNA double helices are right-handed. So you don't necessarily need to know exactly what that means, but there are right-handed and left-handed helixes. It basically just is about the way that they kind of spiral.
Like snails.
Yeah, exactly, yeah. And we kind of spoke about this in the episode talking about the thalidomide tragedy.
Mm-hmm. Yeah.
If you want to understand sort of handedness when it comes to molecules, go ahead and check out that episode. So, the third sort of part of the model is that the DNA double helix is anti-parallel, which means that, we were talking about this earlier, the 5' end of one strand, is joined up to the 3' end of the other strand. So one strand goes 5' to 3', the other strand goes 3' to 5'. Yeah?
Yeah.
Yeah.
I don't know what that means, but yeah.
We got two arrows. One arrowhead is pointing one direction. The other arrowhead is pointing the other direction.
But I don't know what it means in terms of the actual DNA.
Okay, think about it like Lego blocks, right?
Yeah.
You've got the top of the Lego block and the bottom of the Lego block. The bottom of the Lego block cannot connect to the bottom of another Lego block, can only connect to the top of another Lego block.
Okay.
So let's say the top of the Lego block is a 5' end, the bottom of the Lego block is a 3' end. Right?
Right.
You can only add
on one side, the Lego blocks are upside down.
Kind of, yeah. Yeah.
Right. Well, okay. I'm sure that is not how it is, but yeah, sure.
Yeah. If you think about it sort of that way, right?
Okay.
Yeah?
Yeah.
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 So the fourth one and the last part of this model is essentially just about the bonding, right? So there's not just bonds between the bases, the outsides of the sort of outside of DNA is also available for bonding, which is useful for sort of, let's say, proteins that need to replicate the DNA, or proteins that want to modify the DNA or protect the DNA, all of that sort of thing, right?
Yep.
So you've got bonds sort of within the DNA, and the outside of the DNA, the double helix, is also available for bonding by certain things. Those are the four main sort of factors of their model, and those are still sort of consistent today. But they did steal some of their data from someone else, obviously a woman, who then died and got nothing out of that until after her death. It's a classic,
she still hasn't got anything out of it.
They
got $1 million out of it.
Yeah, exactly, right? Yeah.
She got dying.
Well, no, that's what I kind of wanted to talk about. The kind of the issues of, I mean, I guess this kind of falls into the issue of how we view science and scientists. Like, this would not have happened if there was a sort of more collaborative atmosphere, right? If we didn't take scientists and say, you made this discovery, you are amazing. Because, I mean, realistically, if they had taken her data, all they needed to have done is said, clearly, this data is this person's. Yeah. This is where we got this. And then she would have gotten she probably would have gotten the Nobel Prize as well, along with them, because she had contributed so much to their work. So I think it's important to consider, like, I guess really how we view science, which is we view it as just individual people doing great things, but that's really not the case at all. And having that view can actually have an impact on how scientists do their work, because they fully just stole from her.
Yeah.
They fully stole from her because they wanted the glory, presumably, or they didn't see her as, worthy of getting the praise. Like, it's really, really weird to me. It's really frustrating that that's how we view sort of the whole sort of mess of science, of individuals doing great things, you know?
I feel like should there not be a little bit higher standard for the Nobel Prize Committee if they realize they've awarded a Nobel Prize to somebody who ultimately did not declare their sources in science, which is like one of the most important places to declare your sources.
There you go.
Could you not have something that sort of retroactively applies? Like, I feel like if you are up for a Nobel Prize, it's like when I sign a contract, right? If I sign a contract for writing something, or I've had signed contracts for releasing my film, there's a clause in there that says, I declare that this is my own work. And what that does is it protects the company I'm signing to. If it turns out to not be my work, they can be sued, but they can then sue me. And I feel like if Nobel Prize guys come along and like, hey man, we're going to give you a Nobel Prize for your great work. Did you do it all?
Yeah.
and you should be asked that question, and you should say yes. And if you don't say if you say yes and it turns out that that's not true, you should maybe have like some of your money. I mean, they can't take it back. It's not how it would work. But like some mechanism,
or at least add her into,
add her onto the Nobel Prize so she gets a Nobel Prize. Like,
well, she was a woman.
That's not a hard thing.
Oh,
she was a woman.
I'd exclude her.
Yeah.
Sorry, I forgot.
She was a woman. No, I mean seriously though, like that probably did contribute to it quite a lot.
I don't disagree. I'm almost certain. But like now we're a lot further along. We're in 2022, and you know stuff's looking up. It's getting better, and I would hope that the Nobel Prize Committee now have something in there that goes, if you say this is all your own work and you don't declare your sources, not that obviously there are other people who make things and you didn't necessarily know about it, but if it's like, you saw this stuff. Because one of the easiest ways to make money in film, by the way, I'm not going to do this, but one of the easiest ways to make money in film is to write a load of treatments for a load of films, right? Crucially, you then protect them, and then you send them out to every production company you can possibly imagine.
Yeah, because then as soon as they make something that's remotely similar, you just go ahead and
you have like you registered it with the Writers Guild, and you have an email showing that you sent it to them, and you sue them. And the thing about that is you'll win. Like, you just will win. Now, obviously, if they can prove that they were developing it way before, then you won't. But if they can't, then it's very likely you'll win. And that's obviously not the case if you if you can prove that they had access or they saw that thing, you can maybe make the case that, well, maybe it went in subconsciously, or maybe they forgot that they'd seen it and that's where the idea came from, that kind of thing. But even in that case, if you can form a clear link, well, then add her to the Nobel Prize. It's not a particularly big deal. She's dead. They're dead as well now, I think, are they?
Yeah, both of them are dead.
So, like, whatever. No one cares. Just add it to the thing to
They're not going to be offended, are they?
Look at this woman who did some really brilliant work and was forgotten and probably had her work stolen by two men who were then given £1 million. Give her a flipping credit.
Yeah. She actually did work that contributed to 2 separate Nobel Prizes. So it's unbelievable to me that happened in such recent history, you know? And people have wanted us to talk about this for a while. And it's just baffling that there's no protocol for that. Like you're saying, that there's just nothing.
Yeah.
You know, she doesn't get anything really. And that sucks. That really sucks. Especially, I mean, I think this could just be solved by having a more collaborative view of science and having it be less a case of this individual has done this. Like, scientists are really good at working with each other. Generally, they can be quite good at working with each other. But when companies kind of get involved, when it comes to that, it becomes very difficult, right?
Yeah.
And when money gets involved, it becomes like we were like people that were trying to build sort of I think it was models to understand how COVID would spread or something along those lines. Something, you know, that would help in the fight against COVID. And not a single one of them. Oh, that was it. It was the models to diagnose COVID, right? Or to figure out who could potentially have it before testing. You know, those those sort of AI models that they built. None of them worked. None of them were good enough. And so many of them were really, really similar, just like kind of slightly different from each other, that every single, like pretty much, if all of the sort of labs that had been working on it had come together, they could have done something better than they did individually, right? Yeah. And That's so strange to me that we want so much competition, We always want to be the best.
Now, I feel like we're slowly moving towards the C word, Corey.
No, not at all.
And it's not competition.
Well, you got two options, I guess. Like, what that feels like to me is that one thing that the music industry has done really well is it's been able to formalize structures for collaboration, which actually makes collaboration a lot better and a lot safer, which means that musicians can go, it's not perfect, but it's better than, say, the film industry, it's better than photography, it's better than a lot of creative industries. And it means that, at least in this country, and I think in the US and maybe in other countries, essentially, if you go and do some work on a track with someone, you might not get paid at all. You might just collaborate with your friends, but you just write down who's contributed to something. And then if that thing blows up, automatically the royalties sort of are distributed. Now, it's not entirely automatically, but it's much more automatically than, say, in the film industry or in many other industries. And it feels like almost you need some kind of, how you cite papers in other papers and that kind of thing. If you had this sort of formalized web of connections between different papers, different citations, all these kind of things, when someone eventually wins the Nobel Prize or wins some massive thing, you could have this system. Like, okay, so say in the example of DNA, it's not Francis or Crick and Watson who won the Nobel Prize. It was the discovery won the Nobel Prize, and then that money, or that credit, or that kind of like financial award can be trickled down to all these different places automatically. In some way, right? And it's about forming these sort of these webs of attribution that happen automatically, so that becomes an industry standard. And then it's any company. You can still have a certain amount of competition. You can have companies going against each other and fighting against each other in creative competition, kind of capitalist way. But then if something succeeds and it built upon previous things, then the proceeds just trickle down and are distributed automatically. You don't have to go like, oh, I'm going to be a good guy and give some money here. If you're citing something, and then crucially, what's even more crucial to that is that if there was a link that wasn't declared, like in this case, possibly there was a link that wasn't declared, the system, or like people within that system, can form that link almost like a neuron connecting. And then those proceeds are redistributed according to that. That feels like something that could well be built.
Oh, definitely. Definitely possible, because papers are published online and constantly, right? You have the references all there.
Yeah.
And you can click the link of a reference and go back through paper and paper and paper. Like, it's all definitely possible. It's all been digitized. It is totally possible to do that. I just think there is zero incentive. Do you know what I mean? No, I agree with you.
Yeah.
I just don't think anyone would want to do that, because
I think the individual scientists would want to, but it's just that scientists are usually employed by institutions, and those institutions might not want to.
Yeah, exactly.
Because obviously, that's what I mean about the music industry. The music industry has formalized that in with some institutions. They have sort of, to a certain extent, formalized it. So the individual, and the thing about musicians is they're generally not inside of larger companies. They're generally individual musicians, individual music, like players, writers, those kind of things. But yes, you're right that like your Pfizers and your who else is there? Procter and Gamble and all those, they probably are not going to want to do that, because a large part of their business model probably relies on not giving attribution to where it's due.
Yeah.
I don't know. That might be...
Maybe. I don't...
Yeah.
Well, no, I think...
Or not giving financial credit. Like, any large business, or any business at all, really, sort of functions on keeping as much of the proceeds for itself as possible, as it can. And obviously you give royalties where you have to, but trying to keep as much for yourself as possible.
Yeah, what I find insane is that you could do work for one of these companies, and you could discover something, and then the company owns it.
Yeah.
You could make a groundbreaking company owns it.
Yeah.
You'd have put in the work for your entire life, but the company owns it, and it's crazy, because they've said, okay, we'll give you some money, right? Yeah. We'll let you survive, and we'll give you all of this stuff that you can't afford yourself. Now, if you do anything major, we're going to keep it. That's the trade-off that you get.
And that's the thing, is you eventually could reach a world in which, because essentially what you're doing there, if you work for less money, for the potential reward, you're becoming an investor, right? You're investing a portion of your time in a potential reward. and you are taking on more risk. And large companies take on large amounts of risk relative to their employees who take on very little risk because they don't have anything else to do. They want to work for a company. They want to do science, and so that's what they want to do. And so the company takes on the risk. If people eventually were willing to take on more of their own risk, say, for example, for 2/3 of the salary, they take a pay cut, but then if there are proceeds from their work distributed through this system that's done automatically, then they get a higher upside.
I think the issue is that this sort of like, oh, we'll take the risk so you don't have to. It's not necessarily a fair sort of, it's not a fair trade there, because you could say, oh, well, you could take on more of the risk by taking a cut to your salary. But generally the reason that people will accept that, like, you know, oh, I'll take a salary and then not own any of my work, is because they don't really have a choice. You know what I mean? In that you need to pay for your house and for food and all of that sort of stuff. So when it comes to a choice between getting money or getting no money, you have to choose money. You pretty much always have to choose money when you're given the option of money or no money.
Yeah, I think there are an awful lot of scientists who've earned enough that they could take on more risk themselves. What I mean is there's no mechanism there for them to even do it even if they wanted to.
But then you'd say take on more. This is the thing. You know, I think that is absolutely the wrong way of viewing sort of science and scientific discoveries and all of it. This is I guess an issue I have when it comes to sort of like companies getting involved in that sort of stuff. They're just not super compatible. Do you know what I mean? When you talk about thinking about risk and profits, you could study something for, you know, your entire lifetime and not make a groundbreaking discovery that makes a lot of money, right? Yeah. You could discover something. You could discover many things.
That's what I mean by risk. The company takes on the risk that you might do nothing of a groundbreaking level when they employ you. Do
nothing groundbreaking level in terms of profit.
Yeah,
but then
that's what I mean. I'm talking about risk purely financially.
Sure, yeah, but this is my point, that viewing science and scientific discovery and scientific achievement and all of that through the lens of profit and risk is just, I think it sidesteps like the actual point of making these discoveries, right? It says the ones that are the best, the ones that are the most important are the ones that will make the most amount of money, which I don't think is necessarily the case. Like, you know, for example, specifically discovering the structure of DNA doesn't lead you to a lot of money necessarily.
Well,
but it does make you then able to understand DNA to the point of, oh, well, now we could splice genes. Now we could work with DNA. Now we can make genetically modified, or like all of that sort of stuff. Sure. But that discovery in and of itself is not like a moneymaker. You know what I mean?
It would be a moneymaker if you were to have this sort of web of attribution where if anybody used your process in some way, essentially like a patent, if anybody used your process for anything, there a small amount of credit, and then they made money off of that. So like if somebody discovers the PCR method and then loads of people sell PCR tests, a tiny amount of attribution goes through that sort of web and back to you or the company that discovered it. So I think when you're talking about, like, so for example, one of the things we talk about a lot in the sort of pharmaceutical method of sort of organizing resources for science is like, okay, if a company discovers a cure to something that it sells a subscription service to, as happened with the stomach ulcers story we talked about in which episode, Jen?
Episode 1.
No.
Episode 1. Great. Yeah. That was an easy one. If you have a very good business model selling, for example, for an easy
treatment, not a cure.
Yeah,
specifically.
Yeah, a treatment. Say for ease, let's say paracetamol, right? Mm-hmm. Like it's especially in some countries where paracetamol or ibuprofen are still patented, and so 10 times more expensive than we have it in the UK. You have the problem, which is pain. And you're selling essentially a subscription service to relieve pain, which is paracetamol or ibuprofen or whatever else, medical heroin, whatever. And then imagine, which you wouldn't do, you found a cure for pain, which you wouldn't do because otherwise you wouldn't have actually helpful pain, like banging your knee or falling over or like you're getting electrocuted or whatever.
Knowing when something is wrong.
When something's wrong, right? You might not want that to be out there because it destroys your paracetamol business.
Yeah.
But the sort of solution to that is governmental awards for the solution for that in an economic system, in a capitalist economic system, is creating financial awards for those who discover things that are generally benefiting humanity, even if they do not benefit a company. And so I feel there is a patch. There are ways to patch capitalism to fix the bad incentives that it creates sometimes.
You've hit the nail on the head.
Yeah. I brought up capitalism, damn it! I can't even do it.
Yeah, no, I specifically was not saying that. I was waiting for you to bring it up. So thank you for bringing it up.
He was leading you into it.
That was the C word, by the way.
So, now that I've walked you into that, yeah, you've hit the nail on the head there, Luke. Absolutely. Capitalism is just not... No, but my point is that capitalism isn't really compatible with the way that we kind of view science, right? Or how science sort of, I guess...
Ideal.
Should be.
Yeah.
Yeah, because when you look at sort of knowledge through the lens solely of profit, then it becomes difficult because you can only if profit is your only incentive, well that's kind of not necessarily antithetical, but that's not compatible with the sort of incentive of science, which is knowledge and understanding and helping people potentially as well, right? When it comes to just like, oh, how can we make money? How can we make the most money? Yeah, you literally have companies that would want to silence and get rid of a cure for stomach ulcers because they're making bank selling the basically the Band-Aid treatment of antacids, you know? Like it's messed up. And you also then don't have enough research going into certain areas because hey, it might not make a profit. There's a risk there. But realistically, when it comes to science, if you're looking at, say, creating vaccines, if you're looking at, say, creating any kind of medicine, there is an incredible amount of risk because it might not be profitable because any avenue you go down could be a dead end. And
that does happen with capitalism. Companies do invest on possible things and then they lose their money.
Yeah.
And that's how companies are selected for or deselected for. But I guess it's almost like I wouldn't say that science is entirely incompatible with capitalism. It's just that it skews it towards certain types of science, and then you have to sort of have governmental agencies prop up the rest of the science.
When I say that it's incompatible, it's incompatible because you need a sort of not necessarily an outside force, but it's not something somewhat external
pure
to come
capitalism where governments don't exist and the free market is the only thing that exists and there are no rules.
Less regulated.
Exactly.
You specifically need to regulate capitalism because it's not compatible.
And I think that's the thing that people really don't understand is that we don't have capitalism because we have on top of it all these sort of patches, like I said, to capitalism, which is like regulations, like, okay, right, you're not actually allowed to do that. Oh, and by the way, you can only charge this amount maximum for this, which
you've got to pay people at least this at minimum. We
don't have it because there isn't such a thing as like a free labor market because you can't choose to not have money because you can't choose to not have food. So there's we don't have a truly free labor market when some of the things that capitalism produces are necessary for existence.
And there's no way to specifically, the key here is that there's no way to access those things without engaging with capitalism
unless you have money already.
Unless you've got capital, in which case
well, you could, unless you you grow your own food on your own land that you already own.
Like if you want to look at the origins of capitalism, that could have been the case, and then capitalism made sure that was never going to be the case.
So, but I always find that very interesting. Is that I think a lot of people think we have capitalism, whereas actually we have capitalism that we keep on adding fixes to. Because it doesn't actually work anywhere near perfectly. So anybody who claims to be a capitalist is like, oh, I'm a capitalist, but I'm not really, because, well, do you believe in the minimum wage? Oh, yeah, I believe in the minimum wage. Do you think governments regulate things? Oh, yeah, well, I think governments regulate things.
I've seen people that say that we should have completely unregulated capitalism. That's the way to go. And I'm there's a reason that we have certain law. We already have issues with minimum wage. And if you look at the US, where waiters are paid below minimum wage, and what's the fix for that? People need to give them extra money so they
mandatory
don't starve.
tips.
Oh, so charity now.
Yeah.
Okay.
Yeah.
Right. So that's not capitalism anymore. Well, it sort of is.
In a functional system, charity would not exist.
In the sort of economic system, people giving tips to people who are underpaid is people being, oh man, the way you would describe that economically, it's hilarious. It's like those people are making, restaurant goers are making the free economic choice to give a tip in order to avoid the negative stimulus of guilt. That's how you would describe tipping.
But also, let's say people were just fine with guilt.
Yeah.
Because what is happening here is that companies would then be weaponizing guilt in order to increase their profits.
Yeah.
Or cut their spending and thereby increase their profits. Now, let's say that people all of a sudden were just like, okay, no guilt, I don't care.
They
would all immediately just collapse because the waiters would either try to find other jobs, or realistically they wouldn't be able to find other jobs because it's not just as easy as finding another job. They would just die, and then who would come in to replace them? Do you know what I mean?
Yeah.
You know, it doesn't. Oh, it's baffling. It doesn't work. It's so annoying. But,
yeah. Who would come in to replace them would be people who are so desperate that they have to just take the wage,
and then they would die.
Yeah.
No, that's what I'm saying. Like, you don't have an unending labor force. At some point, the working class is going to die out if you're not paying them a livable wage.
Oh no.
Well, I'm just.
Or they're either going to die out or they'll revolt, and that's probably what they'll do. Is they'll revolt and then they'll kill you, and then you get a revolution, and then they set up a government. And then they go, oh, what was wrong last time? Oh, these things were wrong. Okay, let's make laws. And eventually you get back to not capitalism again.
Yeah. But capitalism is, I think it's fair to say that it's not really necessarily sustainable. So this is what bothers me about people that say, oh, I like capitalism. You kind of touched on it. Like, you've never seen capitalism in your life. Like, You don't know what that's like.
How capitalism do you want to go?
Yeah, exactly. You know, if we leave the free market to decide stuff, people die. That's pretty
capitalism with all the band-aids on it.
Yeah, it's like, oh gosh, this great boat that I've got, it's amazing. Oh, it sprung a leak there. Don't worry, I'll plug it up. Oh, there's another leak. Oh, I'll plug that one, and then the entire boat is just like corks that you've plugged the leaks up with, right? It's like, your boat is no longer there.
I love my boat.
I say we just plug all of the leaks in this capitalism boat until we've made it entirely out of socialism. Then we fixed it. We fixed the problem.
Oh no, I love capitalism. Capitalism's great.
So funny.
But it's so interesting to me the way that people think of I was speaking to someone the other day that thought that the UK was socialist. Because
do we have the NHS?
Well, this is what I was saying. I was like, every country you kind of know of is capitalist.
Yes.
More or less. Pretty much everyone you'll have heard of is probably going to be capitalist. Just because they have kind of socialist policies does not make them socialist countries. They're still capitalist at their core. It's just they've just got some regulations.
Yeah. It's got bigger band-aids on them.
Yeah. With capitalism, there would no free healthcare. You pay for healthcare. You've got lovely insurance companies which will then give you healthcare and they make a profit. And you have the choice to either have healthcare or not.
Yeah.
You have the choice.
Yeah, the free choice.
Now,
that's freedom.
If you do that, you end up with America, which objectively sucks. Yeah. Or you could have a lovely nationalized healthcare service where things are quite a bit better.
And you spend less pounds per person on healthcare.
And you wait longer. Oh, so much waiting.
57 hours to see a doctor.
Yeah, except you can also just call...
my taxes are $3 billion a year.
You could also just call 111,
Oh, yeah.
Yeah.
Do that.
You don't have to pay for that. In the US, you'd have to pay for Call a Doc. Anyway, we're getting a little bit off track. What were we talking about today?
DNA?
Oh,
well,
yeah.
Good. We really turned this into an After Dark, which if you liked this little conversation at the end, check out our bonus show over on our Patreon, After Dark. Comes out every month. But I think that's more or less it for us this week. What do you think?
I thought it was a pretty good episode.
Yeah.
I think this is not more or less this because we have one last thing left.
Yeah, we do have one last thing left. It's a quickfire quiz.
Dun, 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 dun. Thievery edition.
Jesus.
What?
So, the rules of the quickfire quiz are going to be exactly the same as always. I'll ask one question. That's one question between the two of you. The first person to buzz in with the correct answer wins. What do they win, Jamp?
Stolen Nobel Prize.
Goodness me. Where you got that one hidden?
I'm telling you.
Jesus. So, Luke, what is your buzzer?
Hey!
Jamp, what's your buzzer?
Give that back.
The question for you is, what is the name for the structure of DNA?
Hey!
I'll give that back.
Luke, what is your answer?
Deoxyribonucleic acid.
Oh, that's not the name for the structure of DNA.
What? Oh, yay! Oh.
Jamp, do you want to come in for the steal?
So the double helix.
No.
Well done. Congratulations. That is it.
That's lucky because I was also going to say that.
And that is it. Before we go, we would like to thank all of our patrons with a very special thank you to executive producers Glitch Rabbit and Donito. And thank you for watching. You can find the full references for this episode in the description. Subscribe for new episodes every Sunday, and why not leave us a nice wee comment? You can support the pod at patreon.com/SCI Guys, or you can find a god that does it, SCI Guys Pod, on Twitter, Facebook, Instagram, and at SCI Guys on TikTok too. Or you can send us an email at sciguyspod@gmail.com.
That's sciguyspod@gmail.com.
SciGuysPod
@gmail.com. You can follow me at NotCorey everywhere.
You can follow me at Jamkin everywhere.
You can follow me at Luke Cutforth everywhere.
Goodbye.
Goodbye.
Goodbye.